Hi, everyone. I'm very happy to be here. And yes, I am part of the Matter to Life program. And so because of that, I will be speaking to you today about biomolecules, simulations, because that's what I do. And as I was preparing for this talk, I thought it was pretty funny that I would be presenting a biology subject, because when I was in high school, and even still in college, I used to hate biology. But then I saw this thing right here. What you're seeing there, it's a kinesin, it's a protein, it's a cargo motor protein that literally walks through the cell and just brings stuff with it. And I remember when I saw this, and I thought it was fantastic how complicated things like this can be, how complicated the world down there is. And it really struck me, and I wanted to understand the dynamics of the stuff that is going down there. So this led me to study biomolecular simulations of any biology structure I'm really interested on. What you're seeing there is the coronavirus. I mean, I don't have to explain by now what the coronavirus is, but I don't know if you heard that the coronavirus has a spike. And with this spike, it is able to infect the cell. And when we want to target it, or we want to understand what it can do or what it cannot do, its limitations when it has mutations or so, we need to look at this as a movement. Now, biomolecular simulations allow us to do something that most other biology techniques don't allow us to, which is to look at dynamics. So essentially, molecular dynamics arguably is one of the best established techniques out there to study computational biology, and it pretty much uses Newton's equations of motion to study the way stuff moves. And that is a very scary equation there, but you don't really have to understand it all. It's just there for you to see how many letters, how many stuff is there that needs to be tweaked and peaked into having the perfect movement of a molecule. So we have to understand that we're dealing with soft matter, stuff that moves. And for us to be able to use of this technique, for me, in my case, I like to do drug design. And for me to do this, I need to look at the dynamics of stuff. Now, simulations are pretty much essential in the research of biology, and one can get a lot of insight, even from the easiest parts. One can do, for example, visualization on a molecule, and you can do this from your laptop and already get a lot of insight into the problem that you're seeking. But if we want to get more serious, stuff starts getting more complicated, and maybe you'll need, for example, a GPU. So like a gamer's computer, you can use this to simulate a small system, a small molecule in water. But then we have to get more serious sometimes in science, and we use supercomputers, and we have a lot of resources for this. And with this, we can do stuff like study ion channels in the cell, so how does the cell dynamics work? Or even in the Alamos research laboratory, they're now simulating a full gene. Imagine the implications of this, having a full simulation of a gene, billions of atoms being simulated. And what we'll have next, well, we don't know, because we don't know how the next computers will be. We'll have some ideas of how they may be, but we're still not sure. But it is predicted that we would be able to simulate a full cell by 2034, or by 2172, a full body. Now, what this would mean for health research, it's huge. Imagine just being able to simulate how a drug travels through the whole body and what it does to it. But of course, we have to stay to what we have right now with the limitations that we have. But even with those limitations, molecular dynamics have done great breakthroughs in medicine. For example, the HIV, something that struck us all, like in the 80s, it struck this world. But in 1999, someone was able to crystallize the structure of HIV. And then someone else went and did molecular dynamics to understand the movement of this molecule. And with this person predicted how a drug towards HIV should look like and where it would bind, and this led to the building of this other drug called raltegravir, and it was even fast FDA approved, and it went to save thousands of lives. And we're talking about simulations being a great deal breaker in this case. But there are still many, many more challenges to target, and the one that I'm mostly interested on are targeting protein-protein interactions. Now, protein-protein interactions are the heavy lifters of the cell. They are pretty much controlling everything that happens within a cell. And if we want to control a cell at some point, well, we're going to need to control protein-protein interactions. And the way we're doing this is why we're doing this is because we want to sometimes break some interactions, because sometimes when they are not well regulated, this can lead to cancer, neurodegenerative diseases, immune disorders, and so on. So, and the way I do this, one of my PhD proposals is to target protein-protein interactions using cyclic peptides. I could talk for ages about cyclic peptides, but I'm just going to tell you that they're like little proteins. So one can use them and select them to imitate parts of protein-protein interactions, and does have something that competes, pretty much mimicking the way the protein already works, and now we can stop this from happening. So we build a very generic method for this. I like building methods, and for example, if we have a pool of all the cyclic peptides out there, and we have a protein ligand target that we want to target, protein-protein target, we can match our cyclic peptides to it, and then mutate it so that it looks exactly like it. And then we do empty simulations and free energy calculations, which are the techniques that I use, and we crown the best cyclic peptide. And now we have a method that potentially can be used to solve many diseases. Of course, there's a huge process to it, and there's a lot of things that need to be taken into account in specifics, but we build a general idea for it. And we build actually a program, and we tested in 159 protein-protein interactions, where 130 of them had a success, and 86% of them were really good in MD. So we're really excited about this output. We tested it, for example, in the MHC1, which is a signaling autoimmune protein, and we actually found some cyclic peptides. Now, this is a very important protein, and we almost found like 6 putative cyclic peptides that could then be studied computationally, and then experimentally. And then we're now planning to build like a website for it so that people can come and put their molecules and possibly get a drug candidate for their protein-protein. Now, but this won't be possible if I don't have the collaboration that one has in this kind of schools, right? For example, working along with other people and working along with experimentalists and ping-ponging the information around so that we have all a sense of what's going on, and then the chemist telling me I'm doing something completely wrong, and I'm telling the chemist, but we should do this, and that is where we grow. But of course, there's still a lot of time, and there's a long process. So it takes me back to the kinesine, and we should take it one step at a time. And yeah, thank you very much. So that was it for my part. So now it's Emma. Thank you.